not to be confused with horsefly. The housefly, Musca domestica, is a fly of the suborder Cyclorrhapha. It is the most common of all domestic flies, accounting for about 91% of all flies in human habitations, and indeed one of the most widely distributed insects, found all over the world. It is considered a pest that can carry serious diseases. Physical description. The adults are about 5–8 mm long. Their thorax is grey or sometimes even black, with 4 longitudinal dark lines on the back. The whole body is covered with hair-like projections. The females are slightly larger than the males, and have a much larger space between their red compound eyes. The mass of P can range from about 8 to 20 mg under different conditions. Like other diptera, houseflies have only one pair of wings. The hind pair is reduced to small halters that aid in flight stability. Characteristically, the media vein shows a sharp upward bend. Species that appear similar to the housefly include, the lesser housefly, Phania canicularis, is somewhat smaller, more slender, and the media vein is straight. The stable fly, Stomaxis calcitrans, has piercing mouthparts and the media vein is only slightly curved. Life cycle. Each female fly can lay approximately 9,000 eggs in a lifetime, in several batches of about 75 to 150. The eggs are white and are about 1.2 mm in length. Within a day, larvae hatch from the eggs. They live and feed on organic material, such as garbage, carrion or feces. They are pale whitish, 3–9 mm long, thinner at the mouth end, and have no legs. Their life cycle ranges from 14 hours to 36 hours. At the end of their third instar, the maggots crawl to a dry, cool place and transform into pea, colored reddish or brown and about 8 mm long. The adult flies then emerge from the pea. The adults live from 2 weeks to a month in the wild, or longer in benign laboratory conditions. Having emerged from the pea, the flies cease to grow. Small flies are not necessarily young flies, but are instead the result of getting insufficient food during the larval stage. Some 36 hours after having emerged from the pupa, the female is receptive for mating. The male mounts her from behind to inject sperm. Copulation takes a few seconds to a couple of minutes. Normally, the female mates only once, storing the sperm to use it repeatedly for laying several sets of eggs. The flies depend on warm temperatures. Generally, the warmer the temperature, the faster the flies will develop. Aging. Because the somatic tissue of the housefly consists of long-lived post-mitotic cells, it can be used as an informative model system for understanding cumulative age-related cellular alterations. Agarwal and Sohal studied the level of the oxidative DNA damage 8-hydroxydeoxyguanosine in houseflies. They found that the level of 8-OG increased with age of the flies. They also found an inverse association of 8-OG level with life expectancy of the flies. They concluded that their results support the hypothesis that oxidative molecular damage is a causal factor in senescence. These findings are in accord with the general view that oxidative DNA damage, particularly in post-mitotic tissues, is a principal cause of aging. Sex determination. The housefly is an object of biological research, mainly because of one remarkable quality, the sex determination mechanism. Although a wide variety of sex determination mechanisms exist in nature, the way sex is determined is usually fixed within one species. However, the housefly exhibits many different mechanisms for sex determination, such as male heterogamy, female heterogamy and maternal control over offspring sex. This makes the housefly one of the most suitable species to study the evolution of sex determination. Evolution. Even though the order of flies is much older, true houseflies are believed to have evolved in the beginning of the Cenozoic era, some 65 million years ago. They are thought to have originated in the southern Palearctic region, particularly the Middle East. Because of their close, commensal relationship with humans, they probably owe their worldwide dispersal to co-migration with humans. Relationship with humans. In colder climates, houseflies survive only with humans. They have a tendency to aggregate and are difficult to dispose of. They are capable of carrying over 100 pathogens, such as those causing typhoid, cholera, salmonellosis, bacillary dysentery, tuberculosis, anthrax, ophthalmia, and parasitic worms. Some strains have become immune to most common insecticides. Houseflies feed on liquid or semi-liquid substances besides solid material which has been softened by saliva or vomit. Because of their large intake of food, they deposit feces constantly, one of the factors that makes the insect a dangerous carrier of pathogens. Although they are domestic flies, usually confined to human habitations, they can fly for several miles from the breeding place. They are active only in daytime, and rest at night, for example, at the corners of rooms, ceiling hangings, cellars, and barns, where they can survive the coldest winters by hibernation, and when spring arrives, adult flies are seen only a few days after the first thaw. As a transmitter of disease, mechanical transmission of organisms on its hairs, mouthparts, vomitus and feces, parasitic diseases, cysts of protozoa for example Entamoeba histolytica, Giardia lamblia and eggs of helminths, for example, Ascaris lumbricoides, Trichuris trichora, Hymenolopus nana, Enterobius formicularis. Bacterial diseases, typhoid, cholera, dysentery, phylogenic Korchdea, etc. Houseflies have been demonstrated to be vectors of Campylobacter and E. coli 0157, H7 using PCR. Houseflies can be monitored for bacterial pathogens using filter paper spot cards in PCR, viruses, enteroviruses, poliomyelitis, viral hepatitis, etc. Potential in waste management. The ability of housefly larvae to feed and develop in a wide range of decaying organic matter is important for recycling of nutrients in nature. Research suggests that this adaptation may be exploited to combat ever-increasing amounts of waste. Housefly larvae can be mass-reared in a controlled manner in animal manure, thus reducing the bulk of waste and minimizing environmental risks of its disposal. Harvested maggots may be used as feed for animal nutrition. References. External links, Housefly on the UF/AFAS Featured Creatures website, pictorial presentation of life cycle, more about lifespan of a fly, by D. Nan, The Housefly and How to Suppress It, by L. O. Howard and F. C. Bishop U.S. Department of Agriculture Bulletin No. 1408, 1928, from Project Gutenberg. Also see, Stockbridge, Frank Parker. How to Get Rid of Flies, The Way They Swat Them in Topeka and Order Out the Boy Scouts to Slaughter Them. The World's Work, A History of Our Time. XXIII, 692 €701. Retrieved July 10, 2009. Page, Walter Hines. How to Make a Flyless Town. The World's Work, A History of Our Time. XXIV, 176 €179. Retrieved July 10, 2009. A.